Hey everybody, so today I'm going to talk about viral evolution and I want to hit on genetic shift and genetic drift. We'll get into all that good stuff. The quintessential virus for this is influenza, specifically influenza A. So I want to talk a little bit about that to give us some background on the virus and influenza. I mean, it's so influential for humanity that it's such a good virus to study. And so that's going to touch on viral evolution. So let's just get into it. So we need to understand influenza a little bit. Influenza is a really interesting negative strand RNA virus. So we talked about that before. What does it mean to have a negative sense RNA versus a positive sense? And in general, what that means is that a positive sense RNA has RNA that can immediately be translated into a protein using the host ribosomes. Negative sense usually needs to be converted to positive sense first, and then it can be converted. So it needs to be its opposite, its complementary strand. Now, there are some complications there. They're not always that simple. Sometimes you can translate both. It gets confusing, but basically, influenza is really, really interesting. So it has a negative sense RNA genome. And this is from our ostensible textbook that we're using from Virology. So I apologize for the quality, but it is really nice. So the virus itself, you can see it's enveloped. So it's got an envelope here in the blue, and it's got a bunch of proteins. And this is why I like Virology so much, because there's a lot of detail here. So there's a few random things, the M2 ion channel, let some ions in there. There's some special proteins that help it with nuclear export to get the RNA out of the nucleus. So RNA viruses typically don't go into the nucleus, but influenza is an exception, which is really interesting. There's lots of other things too, and this is where I want to kind of dispel, that might be too strong of a word, but some of the rumors, our understanding of viral complexities. So we say that viruses have an envelope and a capsid and then their genome. Well, viruses are more complicated than that, of course. That's like saying humans have skin and muscles and bones. Well, it's obviously more complicated. And so what we see on the inside is that we can say that the genome is encapsulated or encapsidated, but the proteins that actually form the capsule are various and depend on the virus. And there's a ton of them. There can be a couple, there could be a ton. Influenza happens to have a few things that we associate with that, mostly matrix proteins. So that's on this brown looking one here. That's your matrix. And what's really nice about the viral zone is apparently 4 years later, they color coded all the segments. So viruses can either be segmented, they can have segmented genomes. That means they have just different segments of genome, as opposed to one long segment or one continuous genome. So influenza happens to be segmented. It has 8 segments, A and B. So there's influenza A, that's the one that we typically think about. That's what causes most seasonal flus, though this year I think B was circulating. This year being 2019, 2020. Influenza A is typically what we see pandemic strains in because it can perform its reassortment and its genetic shifting in more species than B or C. C only has 7 segments, so it's lacking a little bit of information there, so it can't, it's generally not as infectious. So we don't usually see a lot of influenza C outbreaks, at least not ones that cause severe illness. So we see some other things associated with the encapsulation of the genome. So we see some nucleoproteins. Those are kind of like your histones, sort of. You can think of them that way. They're just nucleo, so they're associated with what the nuclear material, not the nucleus, but the nuclear material. That's where these nucleo things come from. And it's protein. So it just gives them some structure. They have other functions. Now, RNA viruses need to convert their RNA into DNA. So they bring with them an RNA-dependent DNA polymerase. So here it is. These 3 large segments, so PB2, PB1, and PA, these segments, notice there's a lot of complications here with ribosomal shifts and all kinds of alternative splices to give different pieces. So viruses are very efficient. So using alternative splicing, they can actually create a couple different chunks, which is super cool. Anyway, So that's their polymerase complex. So they need to bring their own because the host doesn't have that polymerase. So they need to bring their own, which is kind of cool. What else do I want to cover? Oh, yeah. So that's kind of the guts of it. And we're going to talk about these segments with genetic shift. But to understand influenza itself, we need to talk a little bit about hemagglutinin. So heme being blood, glutenin being agglutinate or to agglutinate. Not gluten like, like in plants, but to agglutinate means to... heme being red blood cells, and agglutinate is when you take a protein, is how this usually came about. You would take a protein or something that you just didn't know what it was, and you would put just red blood cells, usually from like a sheep, and you put those in a little like a little well, and if it agglutinated, they would form up. So they would kind of form like a little, almost like a scab. It's not right. So they would come together and stick. And so that was some proteins cause agglutination and some don't. A lot of viral proteins actually do cause that. And so like when I was a TA for virology at Colorado State, we would do agglutination assays to look at different viruses, and you could do dilution assays to sort of kind of figure out like, oh, what's the threshold of agglutination? Kind of an older school way of actually doing it, but it does teach you a lot about viruses and agglutination and stuff. Antibodies also can perform agglutination. Neuraminidase is the other biggie. So hemagglutinins are yellow one here. It's coded by this sort of the same color segment here. And then we've got our neuraminidase. And so that's coded down here. So you can see it's like a lighter green. This nucleoprotein here is NP. That's these ones over here. So we see we've got our neuraminidase, and we're going to talk a little bit about those two things, hemagglutinin and neuraminidase, because those show up actually in the news. Lots of people have heard of H1N1 or H5N1. That's what these are, hemagglutinin and neuraminidase. So let's talk a little bit about their functions, so that the next time you hear about influenza, oh, there's a new strain of H1N1 floating around. Oh, I know what that is, because that seems like something valuable that you could learn. So here's a typical influenza A. This is from a really nice Nature article. So I have the links all down here, so go ahead and click on those. I tried to pull from really good papers and really good websites for this talk particularly. I mean, I try to do that for all the talks, but this one especially, there's some really good ones, especially the next slide. So here we have a nice Nature article talking about influenza growth and the influenza growth cycle inside of a cell. I want to dig a little bit deeper into this event here and the budding event here, so entry and budding. But just to give us a brief overview, this is going through, I wouldn't say extreme detail, but a lot more detail than you would typically find for a virus where we attach, enter, it goes through endosomal pathways. Entry is actually driven by a sialic acid binding of the hemagglutinin, which we'll talk about in a second. And then we have that endosomal release. I believe it's clathrin-coated pit release. There's lots of different ways that we can actually pull endosomes in and send exosomes out, or send endosomes out. And so if you want to learn more about that, Viral Zone has a really good overview of the different ways that viruses can actually get in and what happens. Remember that a lot of times as we have the endosomal pathway come in, the pH change is actually what allows for conformational changes of the proteins. And so that's where we actually get some of this matrix. You'll notice the matrix protein here, so it's still there, right? The brown matrix. So as we enter here, the pH is going to change, and we know based on the pKa of a protein that if we are in a more acidic environment or a more basic environment, that can change how many hydrogens are attached or how many hydroxides are attached to a protein, which can cause a conformational change. And so we see that with these matrix proteins. Influenza is very strange. It's a negative sense RNA virus that needs to go into the nucleus in order to replicate. That's atypical. Most RNA viruses avoid the nucleus. Most DNA viruses typically go into the nucleus. Now, there are exceptions to both, obviously, because we're looking at one right now. Once it gets into the nucleus, though, it needs to make more of its segments. Now, it has 8 segments, so you need to make enough of those for each new baby virus to bud off. And so just think about the complexity of that. There's 8 separate segments, all different sizes. You need to make sure you have one that goes into each virion. Things get really complicated. If you want to read more detail about it as well, you can go to that Viral Zone article that talks about influenza, just right here, because it does some really, really interesting things with its genome. How it gets its RNA, this is the viral RNA, how it actually gets it to be capped, because we know that ribosomes require them to be capped and tailed, and our RNAs to be capped and tailed in order to translate. And the host's defense machinery will look for that. So it's going to look for 5' caps. It's going to look for 3' tails. And it's going to look for those things and if it doesn't see them, it will degrade it. And so influenza is trying to avoid that problem. And how it does that is really interesting. Sometimes it steals the caps from host RNA and actually puts them on its own RNA, which is really, really interesting. Anywho, so their implication here is that it becomes messenger RNA, and then it starts making, instead of host proteins, they're making all these crazy proteins, which we talked about here. So all these segments then go get translated. And then it has to organize all of them, which is a complicated endeavor. And then it needs to also assemble them and then bud back off. And that's the typical term of use. And you also need to have, your hemagglutinin and neuraminidase. Those are surface proteins. So let's dig a little bit deeper into influenza entry and exit, because that's going to be just something relevant to you, because it's H1N1. I mean, we need to talk about the H's and the N's. So influenza A, when it needs to enter, viruses, well, we've talked previously, they have different tropisms. So some viruses have a very broad tropism. So that can kind of go 2 different ways. So either they have tropism within a host, so they could infect. So HIV, for example, has very narrow tropism for within a host. It needs to have CD4 receptors. So T helper cells have those and macrophages have those. So that's pretty narrow. Influenza, on the other hand, it gets into a host and it can infect a wide range of cells. Now, typically we see that in the upper respiratory tract, but that's probably more a response to how it actually gets into a person as opposed to what it likes. Let me rephrase that. The route of infection is typically respiratory droplets, and so we see them infecting those cells. But they can infect other cells as well, because they don't require just a CD4 receptor, which is only on specific cells. They can use sialic acid, which is on a ton of different cells. Now, we know on the surface of a cell that there are a ton of things on the surface. It's not just a phospholipid bilayer with a few proteins. It's incredibly complex with tons of proteins and glycoproteins and just sugars and massive polysaccharides. Polysaccharides are really, really important for the surfaces of cells. And you can see here, so here, this is on the surface of the host. So this is the cell that this virus is trying to infect. So think somebody sneezed an influenza into your lungs, and this is one of those lung cells. And here's influenza, it's coming along, and it's gonna use its hemagglutinin. So there's hemagglutinin right here. And look at that little pit right there. So that pit is going to fit onto this little chunk right here, or I think it's actually this chunk, of sialic acid. Which sialic acid is just one piece of this polysaccharide here. So you can imagine this specific polysaccharide can be on multiple cells. But what we're worried about is the lungs. And so here's our lung cell, and it can get in, and that's how it gains entry. So viruses pretty much always have a ligand or a surface binding receptor, and then they will bind to a ligand on a host cell, or I might have those kind of backwards. Like a host cell has a surface receptor and the virus is a ligand. But you can see it's kind of confusing. You could kind of use either term because hemagglutinin is technically grabbing onto the sialic acid portion of this polysaccharide. So anyhow, if you've taken biochemistry, awesome. That will be super helpful. If you haven't, then these might look a little confusing, but hey, it's really not that complicated. They're different sugars and sialic acid. I don't know. They're just carbon structures. All right. So, and then it's going to enter, and then you can see I mentioned endosomal pathways having pH be very important. So notice they're mentioning pH here. 5 is slightly acidic. So if your pKa is slightly basic from that, then you're going to start tacking on hydrogens where they wouldn't normally be, and that can cause conformational changes. That's the conformational change that's happening here. So remember, we want to, they're showing here the trans, as it moves into the cell, we actually gain a pH change within the virion itself, which causes more conformational changes. Specifically, it's causing this sort of joining here, which just looks actually very similar to how HIV actually joins itself and blends its phospholipid bilayer with the host phospholipid bilayer and sort of joins up. And so then it releases all its proteins. Now, this comes from a really good Frontiers paper, Frontiers in, I believe, Virology. It might just be microbiology. But it's a really good article. You should look at it because they have other pictures as well. I only just picked 2. So I have this one talking about entry. So hemagglutinin is responsible for entry. That's the most important part of me talking for the last 5 minutes. The actual exit is also very complicated. This paper itself, it's from the same paper, talks about the entire life cycle. So if influenza is really getting you excited, then I would encourage you to go and read this entire paper because it's really, really good. Their diagrams are really good too. I like Frontiers. They make good diagrams because I'm a super visual person. So here we have, we're making all the parts that we need. So we see here they put neuraminidase as these orange little dudes and hemagglutinin are the purple ones. We need to get those onto the surface of the cell so that when the virus buds off and brings its host or brings its genome with it, that it can bud off and then make new baby viruses. And so that's what's happening here. So they're just trying to get everything to the surface so it can bud off. And then it's going to follow these couple, how do we actually get the virus to bud off? That's a great question. It's something that people don't think about. They're like, oh, a lot of people think about the burst concept of like a bacteriophage, which yes, that is a way that bacteriophages get out of cells. They just cause it to explode and then who cares? As long as your virions are constructed, that's fine. But a lot of viruses, especially envelope viruses, are much more complicated than that. And they require a lot of effort to actually get the virion constructed and on the surface and released. So that release is really important. And so we see here, we've got these viruses, they're stuck here. Now remember, what did hemagglutinin do? Its function was entry, right? So it bound to that sialic acid residue on that polysaccharide and it held on to it, right? Well, that's the exact same thing that happens here. So as the virion is trying to leave, that hemagglutinin is still stuck onto that cellular polysaccharide right there. See that? And so what's going to happen is it's going to move that over here to neuraminidase. Now I've seen this portrayed differently a couple different ways. So there's actually probably a more complex, a complex of what's actually happening here, but this is a simplified version. But basically, you need to get rid of that little sialic acid residue, that little red residue there. And so neuraminidase is going to do that. It's a protease. It's going to cleave. It's actually performing water-mediated hydrolysis. So it's a pretty straightforward protease cutting of that bond right there. Can release that sialic acid release, which then allows, that sialic acid isn't there anymore, so hemagglutinin doesn't have anything to grab onto. And so it falls right off. Awesome. This just sort of depicting that same thing. Releasing from cells, getting viruses. Sometimes viruses can stick to each other, so that's something else that neuraminidase can prevent. So you'll see here's a virus with that same polysaccharide, and it's stuck to another virus, so that's not good. We don't want that to happen. And so neuraminidase can cleave that. And then sometimes, so this is kind of cool. You think about this. So mucus is a great way, a great weapon that humans have, and you can take immunology from me if you want to learn about that, a great weapon for trapping viruses and keeping them stuck. But mucus has tons of polysaccharides. It usually have, I can't even talk. It'll have a protein segment with a bunch of polysaccharides attached. And it happens, some of those polysaccharides are going to have sialic acid. And so that is designed to help trap these, the hemagglutinin. But neuraminidase, once again, can save the day if you're the virus and cleave via the same hydrolysis, sialic acid hydrolysis, and allow release of that virus. Awesome. So really, really interesting. So what did we learn? Hemagglutinin is responsible for viral entry. Neuraminidase sort of allows the virus to exit, but we know it's more complicated than that. It's basically relieving hemagglutinin of its burden of holding onto that sialic acid, which allows it to fall off. Now, you may have heard of, there's a couple drugs out there. Tamiflu is the brand name for oseltamivir. There's another one out there, and then there's a couple others that work, and I think there's some in the pipeline as well, that are neuraminidase inhibitors. And so they prevent this from happening. And so what's that going to do? Well, now the cell is infected. That is absolutely true. But all these virions can no longer release. So they're just stuck like this. So you just have an infected virus with thousands, millions of virions just kind of stuck on the surface, which is kind of gross when you think about it. But that's awesome because they're not able to release. Now, they could infect a cell that's right next to it if they could get close enough. So that's potentially possible. But that's how neuraminidase inhibitors work, which is really nice. You can see they would also help with sort of secondarily allowing mucus to trap the virions. So that's kind of cool. All right, so now that we understand a little bit about influenza, I just wanted to cover that since we're going to be talking about influenza a lot. Let's kind of back up, or we're going to shift gears a little bit and talk about the influenza genome itself. So it's segmented. I'm going to back up a little bit. Actually, let's talk about this. So we have 8 segments, and those segments are different sizes, but there's 8 of them, obviously. I keep saying that. And so we sort of have 2 different ways to approach those segments. They're RNA, so they're cytosines, guanines, uracils, and adenines, right? And so we can change their order. We could form small mutations. If you've taken genetics or like a microbiology class or even a basic biology class, you might talk about mutations and what those are. And the accumulation of small point mutations, so just shifting like an adenine to a guanine, or if you're DNA, a thymine to a cytosine, it doesn't matter. Those all have different names, but just point mutations shifting one nucleotide to a different nucleotide can cause small changes over time, and we do see that. That's when we see, if you apply a selective pressure. So in HIV, we see this all the time. You take one drug, you have a patient who has HIV, you put them on one drug, and you're putting a pressure, not a literal pressure, but a figurative pressure on that virus. And so you're saying, hey, virus, if you could figure out a way to avoid this problem, you could keep reproducing. And viruses do that, usually by antigenic drift. So that's where they gain small point mutations, and it's always a numbers game. Remember that about viruses. They're kind of like cockroaches. If you keep using the same brand of Raid roach killer on cockroaches, every now and then you might get one that just happened to have a mutation that allows it to resist that. And so now that cockroach, you can spray with all that Raid you want, but it doesn't die. And you have to switch chemicals or move to something different. Maybe you need to use like a physical destruction. Stepping on it is what makes me think. So that happens in biological systems. We see antigenic drift. Shift is something special. Not every virus can engage in antigenic shift. Most viruses can't. But if you have a segmented virus, especially influenza, then we see antigenic shift all the time. And what that really means is the virus, one version of influenza A, so one subspecies, it's just circulating. Let's say you get the seasonal flu. And let's say you're a farmer and you get the seasonal flu from your neighbor, and because you hung out with him and you said, hey, neighbor, how are you? And he sneezed on you, and now you have the influenza A. It just so happens that it infected this cell, the cell in your lungs. And while it was there, you were like, well, I'm a farmer, I need to go check on my pigs. And so you go and look at your pigs, and one of your pigs sneezes on you too. And you're like, really, pig? That wasn't very nice. You sneezed right in my face. And this pig influenza, now the pig sneezed on you because he has influenza as well, influenza A, and he sneezes on you. And this new influenza virus This should say virus B. That's a mistake. Not by me. I did not make this chart. Hang on. Watch this. Shape fill. Wait. There we go. I apologize. I just missed that. So from a pig, from your neighbor, so it's a human, and if they infect the same cell, which you can imagine is very, very rare, it's all about numbers. So if you have a huge dose of virus from this human neighbor and a huge dose of virus from the pig, then hypothetically you could get them both infecting the exact same cell because the entry is the same. And so they get inside the same cell, and when they actually go to make babies, which is why I wanted to talk about this, they go to make babies and they're making all these babies. And so you have a mixing of those 8 segments, one from the pig and one from the human, and they're all mixing up together, and all the babies can get mixed up. They're all random. And so what we end up with is a baby that can have a few segments from A and a few segments from B. And so now you have a human-pig hybrid, which is kind of crazy. So here's just another visual. So antigenic shift is just, those little point mutations that happen. So in this case, we have H1N1 infecting some waterfowl, and then it can have just a couple point mutations and allow it to infect pigs. Okay, fine. Sure. That's just kind of how viral evolution happens. But what we could do is, in this case, we have humans that sneezed onto a pig that was already infected. It infected the same cell, made new baby viruses, and they had a massive genetic shift. So one entire segment shifted. So here's another visual here. So here we've got our red virus. I like this one because it's just very clear. Unless you're colorblind, then it might not be. Here we've got our hemagglutinin and neuraminidase and all these other segments from a bird. And here's the human, okay, in blue. And they happen to shift. It infects one person. It got lucky. These 2 viruses got lucky and got to be inside the same cell. And I said that really weird, I'm sorry. The same cell. And the new progeny viruses, the new baby viruses that got made, were actually a hybrid. And so you see here, in this case, we have a strange mix. So we've got a couple red pieces, and then we've got some more blue pieces from that human one. So it's more human than avian, but it's kind of a mix. And that's where you get these pandemic strains. So what happens is a lot of times you'll have humans and birds and pigs, and they're all in close proximity to each other. So that happens on farms, it happens in markets. These Chinese markets is what we always used to talk about. That's where a lot of this can happen. Not just Chinese markets either. They can happen a lot of European markets, even US markets. You can track these things. Just when you get all those animals in close proximity, if one gets sick, viruses can just get lucky and actually infect these other species. And so once you have that infection, you have these new progeny, and they look slightly different, but they're still able to infect. And so it's blue enough that it can infect humans still, but it's super red, notice, on the surface. And so depending on what it got from this avian host, it might have a really strong way of, maybe it infects cells really fast, just for whatever reason. Maybe it makes its RNA really fast, faster than a typical influenza. Or maybe it's able to evade some of the host immune response better than either of the previous viruses. And humans hadn't really encountered that, and so we can get that to become a pandemic strain. And so that's where that comes from. Now, if there's mutations within that strain, point mutations, then that's where you get these shifts. And so what they're trying to describe here is there was a pandemic H3N2 influenza strain that came from a host avian complex, H3N unknown, and a seasonal H2N2. And so it basically swapped that neuraminidase 2, it swapped the hemagglutinin out. So it swapped its human hemagglutinin with an avian one, and that just really screwed things up. It also traded some other stuff, one of the replication enzymes, the PB1 from the avian. But then the seasonal flu then, like next year, because it's just kind of circulating in the human, got a shift. And so in its hemagglutinin, which it got originally from that bird, we see a change. And so I really like this because they show it visually. The red hemagglutinin is from the avian, and in the seasonal one, it actually had a point mutation to alter its surface target. And so that's going to change maybe its ability to bind sialic acid. Maybe it can enter more cells. Maybe it can enter them faster. You never know. There's all kinds of crazy things like that. Influenza is really interesting. There's whole fields of study. And so I'm kind of focusing in on 2 different articles here. I like this one a lot just because it kind of shows visually the shifting of the different segments. Really interesting paper you can look at. But this Nature paper that I found is really, really good as well. And it talks a little bit more about the history of different pandemics. So we see influenza pandemics. It's really only been an issue since humans have been able to spread very quickly across the globe. Now, coronavirus we're seeing is sort of just another virus that's taking advantage of this, but influenza is obviously one that we've been dealing with a lot longer. And it requires globalization, which will never go away. And so we can see the 1918 flu was a really bad one, obviously. And what they've done is you can track where the outbreaks started and then when they spread. And notice here, we're talking a lot right now about coronaviruses and secondary waves that show up. And the 1918 flu was really, really bad. It's paralleling this current pandemic really, really closely, which is terrifying because the 1918 was really bad. So you had those first outbreaks in red here. So where's our red outbreaks? So we had a few outbreaks, and then the blue is the secondary outbreaks. And so 1918, we were having people come back from World War I, is basically how this spread. It spread everywhere. It was terrifying. It killed so many people. I previously had mentioned a video. It talked about coronaviruses and influenza pandemics. I will link to it. I will post that below again, just to emphasize if for some reason you didn't watch it last time. But the spread of viruses is really interesting, to cut it short. And this paper will actually go into a little bit more detail about exactly like how we track these things and how we track the current outbreaks. And they're still related oftentimes to the previous influenzas that we've had to deal with. Viruses are never going to go away either, so these things will always continue to happen. It's basically what we've been learning. From that same paper, it talks like the entire pathway. Influenza is really awful and how it actually infects people. And substitute coronavirus in here as well, or any of the other emerging infectious diseases that can infect multiple animals. How we actually deal and manage those infections and the animals, because we can't just cull all the animals. We can't just get rid of all the pigs. That's what stamping out means, is culling, like killing them all. That is one method. You see that on poultry farms sometimes. They'll just cull all the animals because they're like, well, it's not worth it. Even if the chickens do recover, they're not going to be sellable. So just kill them and start over. But you can see some of the things that come up. Water treatment. Basically, it comes from you need to have hygienic animal husbandry. If you don't do that, then you're just asking for viral transfer. And that's what we're seeing. A lot of the, this is not a racist statement, a lot of the Asian markets in particular, though having been to some of the African markets as well, some of the rules that apply, even in America, I mean, not everybody follows the exact rules. And so, like, I don't know, people sneeze on stuff. But the, especially the Chinese markets, some of those are kind of well known for being not super well controlled. And so for whatever reason, you know, no judgment. I don't know. If you don't have the rules, then why follow them? Because it's a lot of extra work. You can make more money technically if you don't have to follow some of these rules. But now we're seeing some of the fallout from this. So if you keep animals in cages and not as well cared for as they probably could be, but that costs money, then you are more likely to get sick. Humans are the same way. You sit inside all day, you don't eat healthy, you don't exercise, you're more susceptible to infectious disease. We know that. And so animals are the exact same way, but sometimes we're caring for the animals. So if you don't do that well, they can get sick, they can get you sick. So, but that's not good. Anyway, so back to genetic shift and genetic drift. So shifting is what we see in influenza. Drift is when we see small point mutations. Okay, those are the biggies. Now, I did want to talk briefly just viral evolution in general. So we know that viruses while considered non-living, still engage in biological practices, if you will. And we see that across all viruses. Viruses have been co-evolving with each other, with living organisms, with us, for as long as you think the Earth has been around. And so we can study that. And so there's entire fields of study that look at different angles of viruses, different genomes of viruses, different protein structures of viruses, and how they're related to other viruses and where they came from. Here's one for lentiviruses, for instance, which just basically HIV, for lack of a better term. Here we see this is a basic lentiviral genome down here. There's a lot of commonalities. HIV shares many of these. Some other lentiviruses have extra proteins, some are lacking some proteins. It gets very complicated. You can see there's a lot of variability. There's consistency, but also variability. And we can track the relatedness of viruses. So in this case, we're trying to track how did these lentiviruses, like where did HIV come from? And you can do that by looking at the proteins and the genomes. And sometimes we want to know, like, where did VIF come from? So VIF is a protein that can sort of protect the virus from the host machinery. It serves that function, among other ones, because almost all of the HIV proteins have at least 2, if not 3 or 4 functions, which is kind of crazy. Where did it come from? Because if we know where it came from, then maybe we could find another animal that is able to defend itself against VIF or whatever. And maybe we can discover new ways of treatment. But you can see here, so here's all the animals that get infected by lentiviruses. There's a ton. So let's see, what are the big ones? SIV, so that's simian immunodeficiency virus. So you can see there's like gorillas. And they all have their own versions too. There's like macaques and gorillas and, I don't know, chimpanzees is a big one. Humans, way down here on the bottom. There are a few different human strains. So you see we share VPU, NEF, VIF, and VPR with a bunch of those. But when we start getting into gorillas, you lose the VPU gene and you switch to the VPX gene. So it's just kind of different, which is really interesting. Why is that? That's a great question. Some people ask that question. Over here we have the feline immunodeficiency viruses. Here we've got some lentiviruses that can infect bovines, some that can infect like ruminants, like sheep and stuff like that. Equine, what is it? Rabbits? Lagomorphs? Cool. Anywho, tracking, looking at these nice family trees, these phylogenetic trees, kind of cool. How you construct different phylogenetic trees is an entire science in and of itself. It's not really within the scope of what I wanted to talk about in this class, but if that's something that tickles your fancy, you can go for it. It gets real mathy real fast, but that's fine if you're better at math than I am. Here, for example, is a different tree construction. Sometimes you see them more like this, but this one actually kind of has both. And this is looking at avian coronaviruses in Egypt. They sequenced their genomes and then matched them by genome relatedness, and you can see how they're related to each other. Some people question that. They're like, well, why do we need to know that? Well, clearly understanding more about where coronaviruses come from could be really, really helpful in how we actually treat them. Because coronaviruses are never going away, clearly. I mean, they can infect all kinds of crazy stuff. Sparrows and egrets, and they like to hang out in birds, but they're in basically every kind of animal. Anywho, so why are these all different? Well, usually point mutations over time, and they evolve within their hosts. And then that sometimes allows them to jump hosts. So hopefully that is a good overview of influenza itself and influenza evolution, and specifically genetic shift. And I'm going to leave you with this picture.